We're up, and we are recording. Everybody say hi. You should. That was me. You can tell your mom because you're on YouTube now. Anyway, so we are going to do just very quickly go through chapter 1 of the book. It is absolutely just an intro. I got so many different machines going now. It's great. And one of the things that we think about when we start talking about science, and I will just say there's some of the stuff, as I mentioned on Monday, that I do PowerPoint-y, and some of the stuff that I'll post the PowerPoint, you'll have these. Other stuff I really like getting back on the board. But science kind of goes in fits and starts here, and it will have these amazing discoveries, and then there'll be quite a range of time where not much is happening. And this certainly is the case in the development of the notion of the bacterium. You know, the idea of single-celled organisms, things you can't see. And so, you know, you have a lot of work going on that finally leads to some kind of great discovery. In microbiology, we have these sort of what we call the golden ages, in which we initially identify germs, animalcules, bacteria, as disease-causing agents. And this leads to a whole study of causes and preventions of a whole range of infectious diseases. I mean, we're dealing with these things right now. You know, we have to remember that prior to von Leeuwenhoek and the discovery of the microscope, the notion that there was microbial life really wasn't something people fully understood. I mean, I can kind of accept this here. As biochemistry starts to take off, which really starts to happen at the end of the 19th century, but really in the very beginnings of the 20th century, we've got the use of bacteria as kind of a model system to start taking it apart and figuring out how things work. And one of the things that starts to happen is we recognize that the same kinds of macromolecules that are in bacteria are in animals, are in us. The same kinds of mechanisms of biochemistry that go on in bacteria go on in us. And so that was, again, a fairly significant discovery. In the past, let's say 50 years, 60 years, as genetics and molecular genetics have given us amazing tools to be able to really take apart bacteria, figure out how they work. We can do recombinant DNA work. We've got a much better understanding now in terms of knowing the amino acid sequence of every protein in a whole bunch of different organisms. And so this is kind of where we stand. And, you know, you think about the fact that we still have pandemics. We still struggle with this. So it ain't like we have the whole story or know how to take care of everything. But we do have some pretty good information here. So, this is kind of a breakdown in terms of some important timelines and things. So prior to 1665, more or less, that is the discovery of the microscope in 1665, which is a fairly primitive tool, but it allowed people to see now at the cellular level. The idea before this was really trying to figure out what caused things to rot, what caused things to mold, what was happening when you take grape juice and you let it sit for a while, and then you start to drink it, and it's like, wow, this is a much better buzz than just drinking straight grape juice. It was not understood. But when people began using a microscope, all of a sudden you start seeing stuff swimming in the water. You start seeing things at a whole different level. And it's like, this is something going on here. So then we've got the pretty long period of time in which the microscope allowed the identification of a lot of these different bacteria and things, but the link between disease and bacteria still wasn't being made. And so it takes quite a while to get to the era of Pasteur and Koch, which we're going to talk about here in a moment, that bacteria and epidemiology, modern epidemiology begins. And so we've got then the idea that bacteria and viruses and things are a source of disease. And so you can then begin to work on figuring out treatments and things. And so this is a bit of a broad swath of 100 years here. But during this time, antibiotics have been developed. The building we're standing in, the Leidy Tabor Science Building, John Leidy was one of the guys that was responsible for figuring out how to stabilize penicillin. And so the discovery by Alexander Fleming of penicillin is a critical part of disease treatment. And further, being able to actually separate out bacteria and modern or standard techniques of microbiology. A lot of the microbiology we'll be doing is stuff that was being developed during this period. So sterile culture techniques, things like that. Then we have the molecular genetics era, where we get to this point where we understand the significance of DNA. We understand the flow of genetic information. That is, DNA is the basic template that then generates an RNA transcript. RNA is translated into protein. We'll go into this in a fair amount of detail as we continue. So this becomes a critical point. And then in 1973, although I thought it was 76, but regardless, the first recombinant DNA molecule was made. And this is done in bacteria, a rather lame kind of project by today's standards, but it was the very first time that people were able to take a chunk of DNA from one molecule, splice it into another DNA molecule, and then put that into bacteria. And so this is the beginnings of biotechnology. And now we've got huge numbers of tools. And we're living in a pretty amazing biotechnology age. The vaccine that we're waiting on is an RNA vaccine, and that technology is absolutely a revolutionary change in how things are working and how vaccines are being generated. So we have the ability to understand the function of genes and the different proteins. We can begin to manipulate things. Think about the fact that we now know that the SARS-CoV virus interacts with the ACE receptor. And understanding that is a critical part to knowing how what tissues in the body are going to be affected by the virus. So this kind of ability is fairly significant. I mean, as you can see, it's not the end all, but we do know what's going on a lot better. So in the pre-microbe area, if we think about this, people didn't know what was causing disease. And if you can imagine people being stricken by fairly dreadful diseases like cholera and plague, the idea that there was bad air and people could certainly smell stuff. We didn't have decent sanitation. The understanding of it, how critical was this? I mean, on the farm, you never really bothered with that. So you don't have the ability to see these bacteria. And so to explain all these things, we've got these different kind of terms. Bad air, or miasma is a term that used to be used. The will of God. So if you don't know what's going on, it can be quite frightening. And plague would rip through, London, through towns, and people wouldn't know exactly what to do. And so you begin blaming everything that you can think of. I'm a big fan of seafaring tales of the Napoleonic War, and the doctor there has his gown that he wears to keep blood off his clothes, but he doesn't change it and never even thinks about it. And he does occasional surgery, and one of the things he does that is kind of tradition from where he was learned was to splash alcohol onto a patient's chest before they cut into it. And they find that people do better and don't really fully understand why. It's just that's how he was trained. The thought that the splash of cold alcohol kind of changed the shock of actually cutting into the patient. But if you had a bad infection, and certainly if you had a bad break that was a compound fracture, they remove the limb. I mean, we don't have any kind of antibiotics. And think about that. You have a bacterial infection, and we don't have any way to treat it. And I don't know, have you guys had bacterial infections? I haven't had one in quite a while, but strep throat, yeah, it can be bad. But I don't know if you've ever cut yourself and you start seeing the red line coming up. That's a pretty serious bacteria infection. And if you don't have antibiotics, you can knock it back. And hopefully you will, but it is by no stretch a guarantee. And so people were kicking off left and right because there are no antibiotics. So this is a fairly primitive period in terms of taking care of ourselves. All right, so the big discovery then, Robert Hooke and von Leeuwenhoek, two different guys, they developed the microscope. Now, these are really very, very simple systems. In fact, you can make, I don't know if you've ever seen some of these little experiments, you can take a little drop of hot melt glue or something like this and build a curved or a half dot and put it on your iPhone and probably have a microscope as good as what these guys had. So, it is a very primitive device, but it does work. The Robert Hooke's microscope was significantly more complicated, and that he had a series of different lenses, and getting up to 1,000x magnification, which is pretty good. That will allow you to see bacteria. So we've got a lot of different microscopes in the building. This is a light these are all light microscopes, and they're limited. Even the really fancy light microscopes are limited to how small a thing they can see, like subcellular type stuff. But at this point, you can begin to see stuff swimming around in the water, and it's like, whoa, what the hell is that? Because you can look at water, and under the microscope, you might be able to see if there's contamination, but in the water itself, you wouldn't necessarily know. So we have this whole unseen world. And from this, they began to draw what they call animalcules. These are very small protists, things like paramecium and maybe amoebas and things. We also begin to see from Hooke the idea of what are called cells. This term cell, looking at cork, and you see this kind of structure here. And the idea was this reminded him of monastery, sort of almost like apartments, and they were referred to as cells, and so that's why we have that name. And by taking a razor blade or a very sharp knife, you can make thin sections by hand and put them on a scope and see all kinds of stuff, particularly in plants. And I've never really done it so much in animals, but in plants you can see lots of things and begin to look at structures and stuff that you would never even conceive of. So the textbook refers to this as the Golden Age 1. That is, the bugs cause disease era. We begin to get these observations here. First off, that and what we're kind of leading up to is modern epidemiology. We see that diseases seem to go from a person to person. So somebody with a cold passes a cold, not some other disease. Specific diseases seem to occur only when specific bacteria are present. That's a pretty significant observation. That bugs, bacteria, seem to come from a previously diseased person. They don't appear spontaneously. This idea of spontaneous generation. And then things that will kill or remove the bugs also limit the disease. This guy, Semmelweis, is credited with washing hands, reducing disease. Now we kind of reflect upon this and think, give me a break. But there was a time where that really wasn't understood. If you start doing nursing classes, you'll learn to wash your hands, and it is a very specific process that you do. but it really helps. It makes a major difference. And so this is the very beginnings of this time of this basically epidemiology. Now, what's really cool is this discovery in 1854 that cholera is caused by contaminated water and not a miasma. And this is really a revolutionary kind of observation in that Snow began looking at cholera transmission. And what he initially did is essentially a Geographical Information System, or GIS, which is a standard tool for epidemiologists right now, is you look to see where people are that get cholera. And then he found sort of the center point and began investigating the location. And he made a discovery of the home with the first victim, had a broken pipe, a suspect water pump, and from there he was able to identify sewer effluent getting into the water supply. And what he was able to do is lock that pump up, and they saw a dramatic reduction in new cases. What he's done is identify the fact that cholera is caused by contaminated water, not miasma, and that is a really significant discovery here. I mean, it's the beginnings of modern epidemiology. So, a couple other examples happening around the same time. You probably have heard of Louis Pasteur, and certainly you've heard of pasteurized milk. Well, Pasteur was interested in looking at the idea of spontaneous generation and basically debunking this notion. Now, we can imagine this, that if you've got a piece of meat and you let it sit out, and after a while, you begin to see little buggies and maggots crawling over it. Oh, yum. All right, you might think, okay, a piece of wood that's dead doesn't show this, but a piece of meat does. There's some sort of life force in this meat. Even though the animal's dead, it's got life force. And from that life force, we get this spontaneous generation of new life. I mean, it's a flawed idea, but okay. Now, what Pasteur did was begin experimenting with different ways of barriers and sterilization techniques. And the thing with the meat is he took this piece of meat and he placed it in a box that had a screen over it, because of course we know, or let me tell you, the reason those maggots get there is that you have flies landing on the meat, laying their eggs. The eggs hatch into larvae, and they start crawling around. And so, you may be looking at it, I won't see no flies on it, or maybe you do, and you wouldn't ever necessarily recognize that that's what's happening. So you put the screen on it, and the meat doesn't get all maggoty. It still rots, but it doesn't get maggoty. All right, so we begin to see that spontaneous generation is probably not something that's happening. The next thing he does is begin looking at things like the requirement of yeast needed to ferment grapes, that bacteria are responsible for souring wine. And one of the things he used was a broth that he began experimenting with, what he would refer to as a wheat straw infusion. You basically go out to the barn and you grab a handful of straw that the animals have been standing around on, throw it into a flask, mix water with it, and you let this sit for a moment. Of course, the straw will settle down. Within a day, this material, this broth, will become cloudy. I mean, duh, it's got bacteria growing in it. What he would then do is build a series of flasks, and he had these gooseneck flasks that he could sterilize, and so he'd fill the whole thing up, take a flame, bend the gooseneck to where it makes sort of a U shape or like a P-trap. And he found that if he heated the extraction, this broth, up to not even boiling, but like to 160 degrees, that it would stay clear for a lot longer. And then he developed, this is in fact pasteurization, and then he developed some protocols where you could heat it up, cool it down, wait a couple of days, heat it up again. and not even to boiling, but like to 150, 180 degrees. And supposedly there are Pasteur flasks at the Pasteur Institute in France that are still uncontaminated. And I think it'd be quite amusing to walk up to one of them and blow into the tip. Because what's happening is that if you have a vessel that's open to the atmosphere, man, there's bacteria coming down. We'll be playing around with this. You take a plate and you sit out on the bench for an hour or so, it'll be covered in bacteria. And if you have this sort of gooseneck, stuff is falling down over here, but it's not making its way into the broth. And by heating it, you kill all the bacteria. And what you do by doing a series of heats, there are sometimes spores that are heat insensitive. You don't kill them by 150 degrees. You'll trigger them to go ahead and sporulate. They'll essentially hatch open. The bacteria that are the form that is now heat sensitive is now that part of the life cycle. And you heat it again, and you knock those guys off. And you do that a couple of times, and this stuff is sterile. So pasteurization, as we'll see in terms of our definition, ain't the same as sterile, but it is a good food preservation technique. You probably, perhaps had the experience of having pasteurized milk that, it's got the sell-by date. Yeah, we ignore that. But if you let pasteurized milk, even completely sealed up, sit in the refrigerator, certainly for a semester, then have your roommate take a big old drink of it and see what happens. It's not sterile. So there is bacteria. The bacteria growth is slowed, but definitely it'll start to go off. So Mr. Lister, who we owe Listerine to, he discovered that there were things that you could use. Carbolic acid, which you may know as phenol, is able to prevent infections. So, one of the things that's happening during this time in the mid-1800s is that chemists are developing all kinds of nasty-ass molecules, things like chloroform, and organic chemistry is exploding. And so phenol is extracted either from oils or made through various kinds of organic syntheses. And straight phenol, man, that's nasty, nasty stuff. But a weak phenol solution is actually a pretty good sterilizing agent. So we began to see that bacteria are disease-causing agents. This is a dude who really discovers epidemiology, this guy named Robert Koch. And he was a plant, I thought he was a plant pathologist, but he did this to a lot of different things. And so he develops this idea that we now call Koch's postulates. And this is, in fact, the basis of epidemiology. And so, start thinking about the pandemic, this is what we're using here. And the pandemic being as infectious as it was, there are a couple of steps that you don't have to do, like step 3. But back in the 80s, when the virus that causes AIDS was not identified, it was a big deal to try to identify the virus, and then, of course, no one's going to inject that virus into somebody. But the notion of how you would stepwise go through the series of logic steps to figure out what is the disease-causing agent. So, like this. You find that bacteria are present in the disease organism. Okay. You isolate bacteria from the disease organism, and you do it in pure culture. And we'll be playing around with this in a couple of weeks, where if you take and swab out whatever orifice you choose to of whatever animal you want to think about, there will be a lot of different bacteria. And what you then do is by limiting serial dilution, you get to where you have individual dots. Pretty much if you were to just spit onto a plate, you cover it with bacteria. We've actually done some oral bacteria studies. There are more bacteria in your mouth, even after brushing your teeth, et cetera, than there are people on the planet by probably 2 orders of magnitude. I mean, there's like billions of bacteria in your mouth. And so you got to dilute it way out to get up with individual colonies. So you get individual colonies and you culture them, and then you take these colonies and you inoculate the little bunny rabbit or mouse, and you see that the test subject gets the disease. That is, you may have a half a dozen bacteria that you isolated. One of them causes the disease. You then see that the bacteria that you cultured are present in the newly diseased guy, and then you can pull out bacteria that's identical to the ones you put in are recovered from the newly sick subject. That is, this is the transmissible agent. So this is Koch's postulates that are used to identify pathogens. And so he identified the bacteria that cause anthrax, tuberculosis, cholera. It's a fairly big deal. And it's just, it's just a set of logic steps. But it is a very specific way of thinking about what is the disease-causing agent. So Pasteur then was doing some of these experiments as well that we talked about with heating things. He also was able to do some filtering experiments and able to identify more or less the size of bacteria. So if a filter can remove the pathogen, it's probably a bacterium. If the filter can't remove the pathogen, it is probably a virus. So viral particles, they're too small to filter. So this thing is really, theater. It doesn't do any good. UV light maybe, but probably not in a fan. So what Pasteur also did is demonstrate pasteurization, the ability to heat fluids, kills microbes, and we'll be talking about some sort of, these definitions. Pasteurization is not sterilization. He also developed vaccines and things. They're using dead or disabled pathogens, and we'll be talking about vaccines a lot too. All right, so antibiotics. These are chemicals that have the ability to inhibit the growth of a variety of bacteria. And we can then treat them with, we can treat infectious disease. Now, one of the things to think about is antibiotics, you have to make sure that they're killing the organism that is causing the disease, but not killing the host. And as we start getting into this a bit further, and you hopefully know this, bacteria are what we call prokaryotes. I mean, they really are way, way different than animals. And so consequently, or plants as well, there are things that bacteria, that will interfere with bacteria, that are not going to do much of anything at all to you. If you've got a fungal infection, or even worse, some kind of parasite infection from an animal, from a eukaryote, they're way harder to knock back because the kinds of antibiotics, quote unquote, that will affect them are probably going to do a number on you. So we're lucky in that bacteria are evolutionarily so distant. There's things that we can do to them that aren't going to have an effect on us. Things like inhibiting cell walls, things like blocking different protein structures that we don't have. So we have various kinds of chemicals that can treat bacteria. Arsenic kills syphilis, but arsenic at too high a dose is also going to kill you. Yo.
Like, what do you think about superbugs, like things that?
We'll be getting into that. It's a scary thing, and we'll be talking about the mechanics of antibiotic resistance. I mean, antibiotic resistance happens because you have a new gene in the bacterium that is able to either detoxify or get around the antibiotic. And so it happens, and it's kind of almost like a prediction that's going to happen. Pretty much any antibiotic you develop, there's going to be antibiotic resistance to it. And there's things like carbapenem, which is one of the very top sort of sparingly used antibiotics that is more or less like the last recourse. And we still have things like methicillin-resistant Staph aureus, MRSA, which is pretty serious in that you get it, and part of it is you start to see what medicine was like back in the Civil War. You had this bacterial infection. There are no antibiotics that will knock it back. What are you going to do? God forbid, amputation? Jeez. So we'll be talking about some of these things as well. Fleming, apparently, the story goes, left the plates out when he went on vacation in London. And when he came back, he had some fungal growth on the plates. And what he observed was there is this area of inhibition, sort of a zone of inhibition, that around that penicillium fungal colony. And so then we have all the normal bacteria. And he says, what's going on here? And he begins actually extracting the fungus and identifying antibiotics. So it takes a while. A variety of different antibiotics are identified. So you can see here, you have a lawn of bacteria, and then you have these little discs that have got different antibiotics. So like this is not a great antibiotic. That is not a very good one. That's barely doing something. But here we got some pretty serious guys. So it's how you begin looking for these things. And one of the things that people do is identify soil microbes, soil fungi tend to have a lot of antibiotics. And people are still, always, identifying new antibiotics. Let's see, purification and sales. World War II sees the mass production of penicillin, and we're off to the races. And so it's been more or less since 1943 that you go into the doctor's office and get an antibiotic. And how many antibiotic-resistant bacteria do we have now? So in under 100 years, we'll probably run out of antibiotics that can actually affect the bacteria that are causing harm. No, hopefully not. So the use of molecular genetics, which is what I do upstairs, that is DNA analysis, gives us all kinds of really cool things. What we can now begin to do is actually get at what's causing one bacterial strain to be really nasty and another strain of the bacteria, same species, to not be nasty. In fact, if you ever look around like the meat recalls, there are E. coli strains that are really, really bad. And then there are tons of E. coli strains that are, you can drink them, you can eat them, there's nothing to them. So there are, in fact, these various toxins, things like cell wall proteins and sugars that they make actually produce that start to screw up your gut. And molecular genetics has been able to show us this. So the different bacterial species and obviously viral DNA is what's happening to infect cells. And there's, in fact, viruses that affect bacteria, what are called bacteriophage, that we talked a little bit about. The repeating helical structure of DNA leads to the idea of how DNA is a replicating molecule. And so here we have James Watson and what's Crick's first name? Anyway, Watson I've met. He's a phenomenally arrogant jerk, but he was standing around when the structure of DNA came around. Francis Crick. And JD can't be with us anymore. They must both be gone. I know Crick's gone. Anyway, they are credited with developing the model of which DNA can replicate, how it's copied. And what they began to do with this structure is you can begin to see how mistakes, that is, mistakes in the copying, are what lead to genetic mutation. So it basically becomes the foundation for all of molecular genetics, which is what we're doing right now. All right, so biotechnology is the latest thing. And what makes this thing work is the fact that bacteria and us share the same genetic code, which, if you think about that, is a kind of amazing thing in that the exact same code for making proteins in us is used in bacteria. So we can do a lot of different things. If you know anybody that is diabetic or you're diabetic and don't know about it, HIPAA regulations. Insulin used to be made from pigs, and since 85, insulin is made in gigantic fermenters is now called Humulin. It is, in fact, a clone of a human gene that bacteria are able to turn into insulin. And there are now tools, pretty easy. You can engineer a little fragment onto the back end of a protein to make it so you can industrially purify that one protein. And so there are tons of now protein-based therapeutics, all kinds of different things. DNA fragments can be cut and ligated and rearranged, recombinant DNA, and since the genetic code is pretty much the same in all cells, we can have bacteria produce human proteins, we can have animals produce bacteria proteins, all kinds of things here. Virtually any gene, you can make any protein, you can put any gene into any organism. We can really begin looking at how these different kinds of specific changes affect the disease. And so, one of the things you're seeing now with the variants of COVID-19, this is coming about through DNA sequencing. And so as different strains come up, and I imagine what's happening is that just all across the country and the world, people are constantly doing sequence surveillance because the virus is not that big, it's very easy to sequence, and looking for individual nucleotide changes that might be significant. The good news about that is that if we can lower the transmission rate, which we're going to do with masks, we're going to do with vaccines, we're going to keep social distancing, by lowering the transmission rate, you lower the mutation rate. And so the notion that we're coming up with these new strains of the virus that may not be, the vaccine may not be effective against, is certainly concerning. The way we're going to get at this is also going to begin dealing with the mutation rate as well. So that is where we stand. And we'll get into some of the next basics of bacteria and things when we meet again on Friday. So, this is the new challenges, that there are emerging diseases. Mostly they are viral. Lyme disease is in fact not a virus, but a lot of viral diseases that are coming out for all kinds of reasons. We also have these re-emerging diseases. Tuberculosis is becoming an issue, particularly antibiotic-resistant tuberculosis. All these various staph infections, sexually transmitted diseases that are becoming antibiotic-resistant. Bioterrorism, God forbid. It's a problem, but in order to deal with this, or to develop weaponized grade stuff, it is not trivial. You've got to really know what you're doing. I mean, it's a lot easier to do a whole bunch of other things than that, I think. One of the things that is happening, and some of this is now getting to be pretty old, but the notion that stomach ulcers come from bacteria is a pretty cool thing. You treat stomach ulcers with antibiotics, not with changing your diet necessarily. We're beginning to see virally induced cancers, and so far in the human population, we don't have things as nasty as like feline leukemia, which is in the cat population, where there's a virus that if you catch, you get cancer. We've got viruses that are associated with cancers, but not the same thing. Heart conditions. Biofilms are another issue that is really cool in terms of like transplants or joint replacements. People will get biofilms, bacterial films, growing on like the hip socket, looks like a Trader Hitch thing, and it can be really difficult to knock back. We're learning more and more about the significance of the microbiome to health. That is, the bacteria that you've got in you and on you. All right, that's all for the day. So we're not going to be doing lab for a bit. I will let you know when we get closer. And that lab will be on Wednesday afternoon at what? I think at 1. So we have time for lunch break. And we'll meet back up. And we'll see how that goes. I think that, at least my view of the requirement is, at this point, everything's got to be able to be online. And so if for whatever reason you're not able to attend the lab and things, we'll figure out some sort of alternate way to beat it out of you. I'm not sure what would be. All right. This is a recording as well. Oh my God. We'll see what this is.